Welcome to This Week in Rheumatology. This week we're covering 8 notable papers spanning early intervention strategies, the fundamental mechanisms of autoimmunity and fibrosis, and new data on lupus therapies. Let's dive in. Our first theme covers treatment strategies, highlighting both a significant success and a notable setback. We begin with a major development in preventative rheumatology. For patients with seropositive palindromic rheumatism, the risk of progressing to full-blown rheumatoid arthritis is a constant concern. A new randomized trial in Nature Medicine offers a path to modify that risk. Investigators compared 2 years of treatment with abatacept versus hydroxychloroquine in 70 individuals with palindromic rheumatism who were positive for rheumatoid factor or anti-citrulline protein antibodies. The study. The primary outcome was the development of persistent arthritis meeting the 2010 RA classification criteria. After 24 months, the difference was striking. In the abatacept group, about 21% of participants progressed to RA compared to 50% in the hydroxychloroquine group. This represents a risk difference of nearly 30%. The time to progression was also significantly longer with abatacept, with a hazard ratio of 0.27. While both drugs were well tolerated, abatacept was also associated with reduced intensity of joint attacks and a higher rate of symptom remission. Conclusions. For this high-risk population, 2 years of abatacept more than halved the risk of developing RA compared to hydroxychloroquine. This is a pivotal study suggesting a paradigm shift towards proactive, targeted therapy in the pre-RA space. In contrast, a trial in a different disease highlights the challenge of improving outcomes, even when surrogate markers look promising. A multi-center randomized trial investigated a novel treatment for adults with osteogenesis imperfecta, a condition defined by bone fragility and recurrent fractures. The study. The trial randomized 349 adults to either standard care or an active treatment regimen of the anabolic agent teriparatide for 2 years, followed by a single infusion of zoledronic acid. The primary endpoint was the number of participants experiencing an imaging-proven fracture. Results. Despite significantly increasing lumbar spine and total hip bone mineral density, the active treatment did not reduce fracture risk. Over the study period, 37% of participants in the teriparatide zoledronic acid group had an incident fracture, compared with 36% in the standard care group. The hazard ratio was 0.97, indicating no benefit. While some quality of life measures did favor the active treatment, the primary goal of fracture reduction was not met. Conclusions. This is a crucial negative trial. It demonstrates that increasing bone density alone is insufficient to prevent fractures in osteogenesis imperfecta, underscoring the importance of bone quality, not just quantity. This strategy should not be adopted for fracture prevention in this population. Next, we turn to several papers exploring the cellular and molecular drivers of inflammation and fibrosis, pointing toward potential new therapeutic avenues. First, a paper in Science Translational Medicine provides a fascinating insight into the mechanisms of pulmonary fibrosis, a devastating complication of several rheumatic diseases. The study investigated why senescent profibrotic fibroblasts persist in the lung. Using single-cell technologies, researchers found that these senescent fibroblasts express HLA-E, the ligand for the inhibitory receptor NKG2A, found on natural killer or NK cells. This interaction effectively puts the brakes on the NK cells, preventing them from clearing the senescent fibroblasts and creating what the authors call an immune-privileged niche. In a mouse model, blocking NKG2A restored NK cell function and promoted fibrosis resolution. Furthermore, the clinical-grade NKG2A inhibitor monalizumab reactivated patient-derived NK cells in vitro, enhancing the lysis of human senescent fibroblasts. This work uncovers a key immune checkpoint in fibrosis and suggests that targeting the HLA-E-NKG2A axis could be a promising strategy to reverse fibrosis. 2 review articles this week put these kinds of findings into a broader context. A review in Rheumatology discusses how pathogenic fibroblasts are not just passive players, but active contributors to tissue damage and inflammation in RA and IBD, and are even associated with non-response to current biologics. The author suggests that directly targeting these fibroblast populations with advanced modalities like CAR T-cell therapy could be a future path to reset the stroma and induce deep remission. Similarly, a review in Nature Reviews Rheumatology details the dual role of innate lymphoid cells, or ILCs, in rheumatoid arthritis. Group 3 ILCs are pro-inflammatory, driving synovitis, while Group 2 ILCs can be reparative. This balance suggests that modulating ILC function, either through cytokine targeting or even cell-based therapies, is another promising frontier. Finally, digging deeper into RA pathogenesis, a study in Annals of the Rheumatic Diseases connects autoimmunity against PAD4 with a specific subtype of RA. The study used T-large granular lymphocytic leukemia with comorbid RA as a model for cytotoxic T lymphocyte or CTL-driven disease. They found that these patients had a strikingly higher frequency of anti-PAD4 antibodies compared to typical RA 60% versus 27%. These anti-PAD4 antibodies were linked to activating STAT3 mutations and a specific CTL memory phenotype. The researchers even detected PAD4-specific CTLs in anti-PAD4 positive patients. This provides a compelling mechanistic link between PAD4 autoimmunity and CTL-driven pathology, potentially helping to dissect the heterogeneity of rheumatoid arthritis. Our final theme this week focuses on refining our therapeutic approaches and diagnostic language, with papers on lupus and autoantibody testing. A post-hoc analysis from the Phase 2 Paisley trial gives us a deeper look at the mechanism of the TYK2 inhibitor deucravacitinib in systemic lupus erythematosus. Methods. Investigators performed whole blood RNA sequencing on samples from over 360 patients treated with deucravacitinib or placebo. They analyzed changes in gene expression over 32 weeks. Results. As expected, deucravacitinib strongly modulated interferon-regulated genes, a core pathway in lupus. But the analysis also revealed novel findings. It decreased plasma cell gene sets and, importantly, increased regulatory T-cell gene sets, suggesting a rebalancing of the immune system. It also enriched dendritic cell populations. These data provide molecular validation for deucravacitinib's action in lupus, confirming its known effects while uncovering new ones that support its continued evaluation in ongoing Phase 3 trials. Lastly, an important consensus statement from Annals of the Rheumatic Diseases addresses long-standing confusion in clinical practice: the reporting of anti-SSARO antibodies. The problem? Historically, anti-RO60 and anti-RO52 have been lumped together as anti-SSA. However, they are distinct autoantibodies with different genetic origins and clinical associations. A systematic review revealed that this confusion is rampant in the literature, with only about 21% of recent studies testing and reporting the two antibodies separately. 16 different terms were used for RO60 and 11 for RO52. The solution. An international panel of 17 experts undertook a Delphi process and reached unanimous consensus. They strongly recommend using the precise gene-based nomenclature anti-TROV2 RO60 feet and anti-TRIM21 RO52 feet. Using this clear language in all future studies and clinical reports is essential to strengthen our understanding of their distinct roles in diseases like Sjogren's disease and lupus, where they carry different implications for risks like congenital heart block and interstitial lung disease. If you only have time for one paper this week, make it the trial of abatacept versus hydroxychloroquine for preventing rheumatoid arthritis. This study provides the first high-quality evidence that we can significantly reduce the risk of progression, from seropositive palindromic rheumatism to full-blown RA, offering a new proactive strategy for this at-risk group. Here are the key takeaways from this week in rheumatology. First, In patients with seropositive palindromic rheumatism, 2 years of abatacept is superior to hydroxychloroquine in preventing progression to classified rheumatoid arthritis. Consider this strategy for your high-risk patients. Second, in adults with osteogenesis imperfecta, a strategy of teriparatide followed by zoledronic acid does not reduce fracture risk compared to standard care, despite improving bone mineral density. This highlights the primary role of bone quality over quantity in this disease. Third, be precise with your autoantibody terminology. An international consensus now recommends distinguishing between anti-TROV2 RO60 and anti-TRIM21 RO52 antibodies, as they have distinct clinical implications. Urge your local lab to report them separately if they don't already. Fourth, mechanistic data from the Paisley trial shows that the TYK2 inhibitor deucravacitinib effectively targets the interferon pathway in lupus, but also has novel effects on dendritic cells and regulatory T cells, supporting its ongoing evaluation. Fifth, emerging basic science continues to point towards pathogenic stromal cells, like senescent fibroblasts, as key therapeutic targets in fibrosis and autoimmunity, with strategies like immune checkpoint blockade or CAR T therapy on the horizon. That's your roundup for this week in rheumatology. The full transcript and references are available on the episode page in your Audio Scholar library. This is an AI-curated summary. For clinical decisions, always consult primary sources and current guidelines. See you next week.